This is Blood and Cancer, the official podcast of MDH Hematology Oncology. I'm the voice of MDH Podcasts, Nick Andrews.
This week on Clinical Correlation, unexpected bad outcomes.
And welcome to episode 60 of Blood and Cancer, the official podcast of MDH Hematology Oncology. I'm joined this week, as I always am, by Dr. David Henry, and people have been commenting on this, but it's so true. It seemed like we just slogged our way through January, and as the days are getting shorter, all of a sudden, this is our first episode in the month of March in 2020. February is gone.
It's amazing. I'm in Philadelphia, and we usually would have had some interesting winter weather in January, February. We had 1 2-inch snowstorm, Philadelphia in the suburbs. I think it was in early January. Nothing since then. We've had 50 and 60 degree days, so when spring comes, it may be 100. So we'll see what happens.
Something is not the podcast to get into perhaps why that is. There are plenty of podcasts that will get into that if you wanted to search your particular podcast app. But we're here to talk about clinical hematology and oncology. This week, the interview is going to be about how to navigate through one of what are millions, it seems like, of clinical trials. We're going to be talking about Keynote, which is Pembrolizumab, of course, and specifically our interview tonight is about how to wade through the statistics. But we'll get into that a little later. First, we want to talk about clinical correlation, which is coming up after our interview with Dr. Ilana Yurkiewicz. And then another heavy topic here from Dr. Yurkiewicz. It's when outcomes for a patient take a left turn out of nowhere, when there are poor, unexpected outcomes. And she said it's impacted the way that she goes over risks with her patients.
Yeah, I think this is a really good one. She does. I often take clues from our surgical colleagues who they'll do an appendectomy, and while they're consenting a patient, you could die. And you think, oh, geez, an appendectomy is no big deal. Well, I think they're better than we are, and sometimes making it clear that while this is probably going to go well, this, that, and the other could occur. And so as we start a new patient on a chemotherapy regimen, or in particular on a clinical trial where there might be some unknowns, we in oncology tend to be cheerleaders a bit. And so personally, I try to be careful not to make it too likely that it's going to be a good outcome and try and be realistic without stepping on the patient's hopes. And so even so, sometimes there's a real surprise and it's not a good outcome. I remember once saying to a woman, there might be some hair loss. She came back 3 weeks later with a bag of hair and said, "This is some hair loss."
Some hair loss, nice.
Sorry, I should have said it better.
Yeah, of course. So that's clinical correlation. It's coming up after our interview this week. For our interview this week, we stay close to home, at least stay close to your home, with a guest from nearby, the University of Pennsylvania, I believe, if that's correct. And we're talking about biostatistics and how to navigate statistics on Keynote 024. There will be 5 digits of Keynote trials eventually, it seems like. But we're going through statistics today.
Indeed. And she is, Lauren Catalano is our doctor who tried to decode and demystify a lot of statistics on this podcast. She's one of our young intensivist attendings. She's really good at that. And she does a lot of our noon conferences with our house staff to go over in journal club situations. Why is this article a good article? And she is so good at explaining and dumbing it down for those of us who have to use statistics to understand articles and data, but don't always know what they're saying. And so what I posed to her, went back, the Journal of Clinical Oncology, JCO, does a most often read download of 50 articles each year. So I went there and found that one of the keynote articles, the Pembrolizumab or keynote trials in lung cancer, was one of those top 50 that came out last year and showed that Pembrolizumab was better than a chemotherapy regimen in this particular advanced non-small cell lung cancer trial, disease-free overall survival. And all these statistical values are popping up positive. So we went through the p-value, the hazard ratio, and the Kaplan-Meier. And so I found it really very interesting, and she makes it really clear and understandable.
It's so important. I remember when I first started in this line of work, it seemed like every day for about a week I would go in and Wikipedia things like p-values, Kaplan-Meier, Cox regressional models, and it becomes second nature after a while. But for someone who was in the literature every day for part of my job, it became second nature. However, if you weren't familiar with able to look at the graphs and go through the conclusions and understand the results, and this podcast, we hope, is a way to kind of be a refresher and also point you in the direction of more robust resources, which are available in the show notes, courtesy of Dr. Ronak Mistry.
Yeah, we thank Ronak, one of our budding some coming soon to be senior residents who's going to go into hematology oncology. Did lovely show notes because the bullet points each week found mdedge.com/hematology-oncology or mdedge.com/podcast. If you want to see some of this in print or review it or click on some links to go where there might be more information, it's all there in our show notes.
Of course.
Thank you, Ronak.
Yeah, Dr. Ronak Mystery. So you can find the notes. There's links, like you said, it's all there. So I wanted to talk about something really quick that this is off agenda for Dr. Henry, but I find it to be a little important. So MD-Edge is owned and operated by WebMD, which is, of course, the parent company of Medscape. Medscape and MD-Edge have a running list of different medical societies and upcoming meetings and what those meetings are planning to do with the current COVID or coronavirus outbreak in North America. In this podcast family, that would include hematology, oncology, dermatology, and psychiatry. Huge meetings for all three of those specialties coming up, including the American Academy of Dermatology, the American Psychiatric Association, and, of course, ASCO later in June. If you have a meeting that's coming up and you don't know what's happening, you can find that list at Medscape and at mdedge.com. And it's one of those things where most of the meetings right now are planning to go on as planned. There are meetings, specifically in medicine and science, that have declared themselves handshake-free. There are flu stations, but there are some, specifically for our European colleagues, that are just going to be canceled. So there's something to think about. That resource is available at mdedge.com and also various places at Medscape.
That's a great resource. No, I didn't know that. I'll after we record, I'm going to go take a look and see any of the meetings coming up as we launch this episode in the first week of March. There may still be. In fact, the epidemic is increasing as predicted, and hopefully it will peter out as we get into April and thereafter. But let's keep an eye on those meetings.
Yeah, absolutely. And of course, if we are covering them, you can kind of follow along with us on Twitter and online, and we'll try to keep you up to date with all of the latest in clinical research. But Dr. Henry, thank you so much. I'm looking forward. I am one of those people that likes the statistics episodes. We do a couple every year, it seems like, and I listen to every word.
I do too. So here it comes, and I hope you all enjoy it.
All right. Thanks, Dr. Henry.
Thank you, Nick. Welcome to this podcast. I'm Dr. David Henry, your host of Blood and Cancer, our weekly podcast, which comes out on iTunes or wherever you get your podcasts from on Thursday mornings. And we are part of the online electronic journal mdedge.com/hematology-oncology, or the same web address, mdedge.com/podcasts, to get copies of this and other podcasts, along with show notes, which 4 of our budding hematologist oncologist residents at Pennsylvania Hospital do nice and dutifully each week. And so today I'm joined by Dr. Lauren Catalano, who is one of our new young faculty, assistant professor of medicine in the pulmonary and critical care division, and an expert, and certainly able to explain to most of us listening today about statistics on something we're going to analyze. So Lauren, welcome, and thanks for joining us on this podcast today.
Thanks, Dr. Henry.
So what I thought we would do, and Lauren and I have talked about this ahead of time, is if you go on to JCO, Journal of Clinical Oncology, every year they say, gee, these are the 50 most read articles. And so I picked one which I thought most listeners are going to know about in treating lung cancer, and it's entitled Keynote 024, Pembrolizumab, which of course is trade name Keytruda, versus platinum-based chemotherapy for advanced non-small cell lung cancer with PD-L1 tumor proportion score of 50% or greater. And the results were wonderful and caused an FDA approval of this. If your PD-L1 is 50% or greater, progression-free and overall survival tended to beat the chemotherapy regimen. But of course, the details were statistical and right over my head. And so I enlisted Lauren to take a look at this paper and address some names like Cox proportion hazard, confidence interval, Kaplan-Meier plot. And as she said right away when we were talking, there's a crossover in here, and there's some primary and secondary endpoints. So Lauren, could you tend to attack the statistics here and dumb it down for those of us listening as to what meant something to you and how you teach house staff to interpret these kinds of statistical terms in a paper like this?
I think a paper like this tends to be really important, but if you don't know why it's important, that's the biggest problem. So when I look at a paper, especially a paper that I will tell you is way outside my general scope of knowledge, the first thing I want to know is what is the primary endpoint? the primary endpoint is the endpoint that's necessary or sufficient to establish any kind of efficacy of your trial, right? The primary endpoint is the endpoint you use to figure out your N or your sample size or how many patients do you actually need to have in this trial to say something that is quote unquote statistically significant.
And that's all done ahead of time. The authors or the statisticians design that ahead of time, and then they accrue to the trial.
You have to be really careful that your primary endpoint is always defined ahead of time, because if it's not, we can think about that a little bit like fishing, right?
And here their primary endpoint, was it the overall survival or progression?
Time since random assignment to disease progression or death from any cause, whichever happened first.
Okay.
So that was really important because death is always an important endpoint, and
It's a hard endpoint.
Hard endpoint, easy to count, easy to calculate, right? You have it. or you don't.
Correct.
So then I look at the secondary endpoints, because sometimes trials can be really interesting because of their secondary endpoints. Maybe their primary endpoint, there was no difference between the groups, but the secondary endpoint will show some sort of clinical signal that might be really, really important to us in everyday patient care and management.
Okay.
So the secondary endpoints mean that these are interesting things that I really want to know about, but I don't necessarily have enough data to say that this is or is not statistically significant. Sometimes it can be, but the study is not powered to see that endpoint specifically.
And what did you see they were looking for here?
So the secondary endpoint was objective response rate,
Okay.
or confirmed, complete, and partial response. They also looked at safety endpoints, which is really common when you're looking at drug trials.
So as we were saying, I think the listeners may well know about this trial and, of course, the FDA approval for this particular drug, but, you know, how we got there. And so the FDA, I'm sure, they went to work and tore this paper apart. So can you explain for some of us what the statistical tests were, and as you're teaching house staff about this in general, what do you say these statistical tests mean and how they're done?
So the statistical analysis, which is usually that paragraph that everybody likes to skip over because it makes them really nervous, is the paragraphs that I focused on the most, because honestly, out of this whole paper, it's probably the thing I understood the best. So what they did here was they took about 170 patients. After 170 patients had died, that gave them a 75% power to observe a hazard ratio of less than 1. And that's usually where everybody stops reading because it's just too much.
Yourself included. So what did that say?
So hazard ratios are important because that's the measure of an effect or an intervention on the outcome of interest, right, which we have here is death, over a period of time, okay? That's all that hazard word throws into the mix when you're thinking about a risk ratio, but now you're going to add time into your equation.
So the hazard ratio is risk and time combined.
Risk per unit time,
Okay.
right? So, and the outcome can be positive or negative, and you have the hazard in the intervention group and the hazard in the control group, and then the ratio of those hazards. So far, so good?
So far, so good.
So then, when you have a hazard ratio, like any other ratio, you need to define the confidence interval. The confidence interval is the range of values that's most likely to include the true population value, because you cannot ever truly measure every single person in the population. You have to have a sample of those people. So
That you hope represents.
you hope, right? And one of the things that makes this paper even a little bit stronger is that it's a randomized trial. So you're going to take people with the same diagnosis and randomly put them in each group. So you're hoping that by chance, the two groups are going to be relatively equal and represent the overall population with the sample.
Okay, and then the intervention and the results. So I'm seeing here, as you point out, the hazard ratio. I see here 0.63 and confidence interval 0.47 to 0.86. So does that mean this was significant?
Correct, because the confidence interval doesn't cross over 1. Pretty straightforward. And when you think about it, your hazard or your risk, right? Hazard is just risk with time. So your risk is either less than 1, greater than 1. But if it crosses 1, if my risk is 1, it's a 50-50 shot of anything happening to me.
So you said for unit time, so of course, probably these metastatic lung cancer patients will all die. So at some point, everybody dies. So I guess with this Cox proportional, hazard ratio, confidence interval, so that the authors or somebody picks a unit of time, and at that unit of time, there's a positive difference for the intervention?
Correct. So a hazard ratio of 0.5 means at any particular time, like I'm going to put just this one point in time, half as many patients are going to have the event as in a different point in time. If your hazard ratio is 1, it's equal. If your hazard ratio is 2, it means twice as many people at that period of time, at that point in time.
And that's bad for the intervention?
In this paper.
Yes, okay, all right. So we have a good news at 0.63, proportional hazard ratio in favor of the intervention, and it's statistically significant, not crossing 1. And then I see lots of curves going on. They talk about Doctors Kaplan and Meyer. Who are these guys, and what's their statistic tell us?
So Kaplan-Meier is just the graphical way that we can put the hazard ratio or a time to event down on paper and look at it to see if we can visually see a signal.
In a graph?
In a graph form.
Okay.
So it's usually drawn as a step function. So if you look at the step function in this paper, each little tick mark is going to represent a patient who falls out at any one given period of time. So for anyone who's looking at the paper, I'm looking at figure 2, A graph, which is at the top, and a Kaplan-Meier curve will always start at 100% because all of the patients are in the trial. So far, so good. Then
I've got it, yeah.
Everybody is at risk. Anybody at time 0 can have the outcome. And they measured and looked at each patient individually over time from 0 months to 33 months. If you look all the way at 33 months, there's no patients left in either group. Because like you said, eventually all of these patients are anticipated to die. It took 33 months in this trial for that to happen.
But as we look at this together, and I recommend to our listeners, because it's pretty straightforward, at 12 months and 24 months, these curves separate in favor of the intervention. Do you think those are statistically different by this graph or analysis?
So it would be very hard for me to say with just my eyeballs if that is statistically significant, okay? So that's why when you look at the number of events and the hazard ratio, which you said was 0.63, which is the number that will likely be toted out of this paper, right, with a confidence interval that does not cross 1, we already know that's statistically significant, but its p-value, and everybody loves to talk about a p-value,
Yep.
its p-value is 0.002, which indeed is statistically significant. Why 0.002? Because in general, we accept a risk of p of 0.05, 5%. I'm going to take a 5% risk that maybe this is not the true answer. Maybe it's 2.5% at the bottom of that bell curve or 2.5% at the top of that bell curve. We add those 2.5s together, you get 5%. That's why P is generally set at 0.05, but that's not a hard and fast rule.
So that 0.05, if we did this experiment 100 times, by chance, the interventions would be different 5 out of 100, but not really so. So we accept 0.05 or smaller to give us reassurance.
True. So this P is 0.002. So you're looking at
Pretty good.
0.2%. Pretty good.
Now, what bothered you when we were talking was crossover. So if we have an intervention which is strong, and we like it, the Pembrolizumab versus the chemo platinum backbone, and this is a positive trial, but they can cross over, do the authors need to define for us when and how, because that'll dirty the result in the control group, now can join the intervention group and maybe do better because of that?
So it's really hard when you're doing a trial on real human beings not to have a little bit of mess, because people change their minds, people change from one group or another. The clinician has the ability to say, well, this is going terribly, we need to change gears, and sometimes that falls in the realm of crossover. This paper specifically says that efficacy was assessed with the intention to treat population. That term is really important because it means that all the randomly assigned patients, no matter what happened to them in real life, were maintained in the group in which they were randomly assigned for the statistical analysis.
So that intention to treat, I've heard you say, and good statisticians like you, that's really good, but I might intend to treat you and you never got it.
True.
And so you're still in the intervention group, even though you said you signed and then a week later, I don't want it, and you left. You're still counted there to be as clean as possible.
You're still counted, and how the authors or the statisticians the authors work with count those people can be more conservative, as in you're in the study for 1 week and you decide not to be in the study anymore, then they're going to count you as censored or falling out or died at that point. That's a very conservative model where it will appear as if more people have died sooner, okay? Whereas you could say, if you opted out on your own volition, you could be censored at the end, which is 33 months. And that could make the study look very, very different. So intention to treat actually makes the study look more like real life and something we can actually believe.
Okay. So we've talked about the Cox, the hazard ratio, its p-value, and the Kaplan-Meier visual depiction of that. And in this case, at 12 and 24 months, the curves are still significantly apart. Primary and secondary endpoints, and the crossover may be dirtying the study a little bit. What did you think of this paper overall, and did it achieve its 50 best articles of 2019 status?
Well, I think one of the reasons, just to go back to crossover for 1 second, that the paper was very strong is that the Kaplan-Meier analysis in Figure 4 actually adjusts for that crossover. Now, there was a lot of fancy math that went into that, and that is Figure 4 at the bottom of page 543. So when you look at that, now there's 3 lines instead of 2. You have your original Keytruda line, standard chemotherapy, and now you have this crossover adjusted chemotherapy. Okay? And the number of events in each one. The reason it makes the paper stronger is even in the crossover adjusted group with 96 events, your hazard ratio is 0.49, and that too is statistically significant.
So it doesn't cross 1.
Correct.
Okay.
So I'm not going to get into a lot of the fancy math that goes into this, but I'll tell you
Bless
that
you.
It will rank those findings and look at the probability of censoring people inappropriately in 2 different stages. And they do a log normal because there's no negatives. Everybody has to be positive. Everybody's on the positive side of the 0, so that's why you see words like log normal used in papers when you can't have a standard bell-shaped distribution around 0.
Okay. All right, so I think we've pretty much taken this paper apart. We like it. We think it deserves its high quality, high flyer status, and actually it helped get the FDA approval for this. And I'll give the title again at the end so our listeners can go look at it if you want online. But a question to you, you're so good at this, and meanwhile you're a pulmonary intensivist and teach our house staff about statistics. Any place you might send our readers, a nice book you like, a primer, statistics for dummies, or websites you go to? How do you keep current or gain all this knowledge?
I think I don't have one single source, is the truth. And it really depends on how much you understand to start. And if all you really get when you pick up a paper is look at the primary endpoint, is there enough statistical power? Do they have enough patients to make any kind of statement? And then understand, is this a categorical value? Does everybody fall in a different kind of pot? Or is it a continuous value, numbers over time like we have here? Those are really the key points. What I would implore to the listeners is that don't just skip over that paragraph that says statistical analysis, because the more times you read it, the more words you will actually understand. And the way I honestly go about it is I read that paragraph until I hit the words I don't understand.
Okay.
And then I put those words, and these were like this rank preserving structural failure time. I had no idea what that was. And I put it in the trusty Google, and I honestly look at the teaching that's out there, not the statistical level journals, because that talks way above all of our heads. But there are good resources out there from different epidemiology schools that can give you 1 or 2 slides and sum it up for you.
All right. Well, I think we've really run through this article nicely. To remind our listeners again, title is Updated Analysis of Keynote 024, Pembrolizumab, trade name Keytruda, versus platinum-based chemotherapy for advanced non-small cell lung cancer with PD-L1 tumor proportion score of 50% or greater. And it was very significantly different in favor of the intervention, Pembrolizumab, instead of the chemotherapy, giving our listeners and our treaters an option of antibody only and no chemotherapy. We referred to this maybe about 2 months ago. You can see online in our show notes an interview with Jack West, who is a lung cancer specialist, was high on this paper and this article and this treatment as well. And I've been talking today with Dr. Lauren Catalano, one of our wonderful young, new, aggressive attending pulmonary intensivists who somehow manages to have a life and be a mom and know statistics, which I'm in awe of. And you can follow us online at mdedge.com/hematology-oncology or mdedge.com/podcast. And there'll be show notes on the webpage if you want to visit us there about today's discussion and bullet points on how to analyze an article like this and do statistics. Lauren, I want to thank you very much for joining us today to help us clinicians and treaters understand statistics. Thanks so much.
Thanks, Dr. Henry.
And that concludes the interview portion of our show this week. Don't forget there are robust show notes available wherever notes are found in your app. Coming up next, Clinical Correlation with Dr. Ilana Yurkiewicz. Welcome back to Blood and Cancer, the official podcast of MDH Hematology Oncology. I am Nick Andrews. It's time now for Clinical Correlation.
I'm Dr. Ilana Yurkiewicz, and this is Clinical Correlation, a segment about the human side of hematology and oncology care. Some bad outcomes are expected. In hematology and oncology, we are caring for critically ill patients all the time. We do our best to try to optimize the chances of a good outcome, but we know that our treatments and interventions are limited. Part of getting used to this field is knowing to expect bad outcomes, even if we do everything right, for a significant subset of our patients. This is hard enough. Then there are the unexpected bad outcomes that I'm sure every one of our listeners who's a provider has experienced. The patient who unexpectedly codes and dies after a transplant, or deals with horrible graft versus host disease, or has an extremely toxic, permanent disability to the chemotherapy that was supposed to help them. These are much harder to grapple with. It's easy to remember our worst possible outcomes because they are heart-wrenching and affect us so deeply. For me, these outcomes have changed how I consent patients up front. I noticed it recently because I think I'm scaring patients more now. Recently, I was speaking to a 70-something-year-old man about what a bone marrow transplant would entail. He came to this visit to hear about this therapy that could cure him, and I spent a lot of time talking about all the ways it could harm him or even kill him. I know this was not what he expected to hear, as evidenced by his reaction. "I think I need a drink," he said. But as I told him, I feel like I'm being more honest, and I don't want to sugarcoat what this involves so that you can make the best possible decision for yourself. I definitely don't want to be skewed by a few bad outcomes, and I of course don't want to scare people away from treatments that I think are best for them. But these unexpected bad outcomes are the cases I will remember for the rest of my life. In hematology and oncology, we are dealing with fire. These bad outcomes are real, and our treatments that are supposed to help can and do cause death or serious harm. I think how I've consented patients about certain therapies has already changed a lot and will continue to evolve. It's impossible to talk about every possible bad thing that could go wrong, but I'm erring on the side of being as comprehensive as possible, even if the chances are low. Because we owe it to our patients to be honest about the good and the bad. Thanks for listening, and I hope you'll tune in next time for Clinical Correlation.
And thank you to Dr. Yurkiewicz for the human side of hematology and oncology care. It's time for this week's credits. Blood and Cancer is produced by the editor-in-chief of MDH Hematology Oncology, Dr. David Henry. It's produced by executive editor Mary Ellen Schneider. Dr. David Henry was our host this week. The Clinical Correlation segment is written, recorded, and produced by Dr. Ilana Yurkiewicz. All MDH podcasts are produced by our editor-in-chief, Dr. Ivan Aransky, and executive editor Kathy Skarbeck, along with multimedia editor Terry Rudd. I'm your audio engineer, audio editor, and the voice of MDH podcasts, Nick Andrews. This